Hello guys, welcome to my channel. So as you know, we are studying about the protein function, and in the last video if you saw, we studied about the myosin and actin, the proteins of muscles. So if you saw the previous video, so we discussed the introduction of this myosin and actin proteins, and further moving in that now. So now we'll see how the muscle contracts, how this too helps in the contraction of the muscle. So the interaction between this actin and myosin, like that between all the proteins and ligands, involve weak bonds, right? And when ATP is not bound to the myosin, a phase on the myosin head groups bind tightly to actin. So I will draw a picture. So for example, so as we studied last time, like actin is a monomeric, right? And it is arranged like this, and myosin is much like this. That's the myosin picture separately, right? Something like this. And the myosin head, like myosin has this head, so myosin head is like this, for example. So this is actin, this is myosin. All right, so this is how it is arranged in the muscles. So when ATP is not bound to myosin and this head, so phase is called as a phase as well, or the head of the myosin, these groups bind tightly with the actin when the ATP is not bound. But when the ATP binds to the myosin and is hydrolyzed to ADP and the phosphate, a coordinated and the cyclic conformation changes occurs in the myosin. So conformational changes occurs in which myosin releases the F-actin subunit and binds to another subunit farther along the filaments. So the cycle, this is the entire cycle. Uh, Myosin head binds to the actin, ADP is introduced and hydrolyzed, and then again myosin bind to the head. So this is the whole cycle, and this cycle is divided into 4 steps. How many steps? It's divided into 4 steps. All right, and in this 4 step, how the muscle contracts, we will see in this 4 step. So in the first step, ATP binds to the myosin, right? So I will draw the diagram again. Okay, something like this. Right, so in this step, ATP is binding. So I will write this down here. ATP binds to myosin. So this is myosin, right? And here ATP that binds to myosin. So when the ATP binds to myosin, what it does, it dislocates the myosin head from the actin. So you can see the space here. So what happened in the first step? ATP is binds to the myosin head, and a cleft in the myosin molecule opens, disrupting. What does that the cleft do? So the cleft is connected to this actin. So when the ATP binds, the myosin molecules cleft opens and it disrupts the actin and the myosin bonding, right? And the myosin is released, as you can see in the step 1. All right, so after this ATP, so in the step 2, ATP is then hydrolyzed, right? And when the ATP is hydrolyzed, it causes conformational changes. All right, so let me draw the diagram again. Okay, something like this. So ATP is still binded to this myosin head, but it is hydrolyzed. So it has now ADP plus the phosphate, and I will write it down. ADP hydrolyzed. So when the ATP is hydrolyzed, it causes a conformational changes in the protein to a high energy state, right? As the ATP is hydrolyzed, energy released. So that energy that moves the myosin head and changes its orientation. So for example, the ATP, the myosin head is bound to this F-actin subunit, but due to this hydrolysis, what happens? The myosin head is now moved, for example, and draw with this. So and it attaches to here, maybe to this unit, to this affecting unit, maybe, right? So what happened in this step 2? So ATP is hydrolyzed, that causes a conformational changes in the protein to a high energy state that moves the myosin head and changes its orientation to the actin thin filament, right? So the myosin, so because of this, the myosin then binds weakly. It will bind weakly to the F-actin subunit closer to the Z disc than the one just released. So it was attached, for example, on this side, but because of this, it goes further and attaches to the another F-actin subunit, but it's a weak bond or weak interaction. So after that, in the step 3, as the phosphate product of ATP hydrolysis is released, right now it has ADP and the phosphate. In the third step, the phosphate is also released, right, from the myosin in step 3, and then another conformational change occur. So what happens in the state 3? We'll draw again. The same actin and the same myosin. All right. So now this is here and the actual head is here, right? So what happened in this state 3? So now, as the phosphate product of this ATP hydrolysis is released from the myosin in this step 3. So what happens here? I will write phosphate, all right. Phosphate is released. In this step 3, phosphate is released from this, so like it has ADP and phosphate. So now, as the phosphate product of this ATP hydrolysis is released in the myosin, right? So another conformational changes occurs in the myosin, and the cleft, that the cleft is open, was open, now it closes, and that strengthens the myosin actin binding. And this step is followed immediately or quickly by the step 4. Right, so you can see that. So for example, it was on this side, right? So that will come back here, but it's here. Alright, so in this step 4, so as soon as the phosphate is released, the myosin cleft is closed, and this process quickly goes in the step 4, that is due to this a power stroke, during which the conformation of the myosin head, what it does, it was here, and due to that it quickly the myosin head returns to the original resting state, and its orientation related to the bound actin changes so as to pull the tail of the myosin toward the Z disc. Right. So in this step 4, a power stroke during which the conformation of the myosin head returns to the original resting state. So it will come back again, right? And in this ADP is released, right? So in this step, the ADP is released, right? So in this step, ADP is released and the cycle will be complete, right? So it will come back to the first stage, right? And each cycle, so this was a one cycle. Each cycle generates about 3 to 4 pn. That's piconewtons. That's a measurement of force. That force, and that moves the thick filament 5 to 10 nanometers relative to the thin filament. Right. So this was the cycle of muscle contraction. So actin and myosin. So the myosin head is bound to the acting in the resting state. When the ATP binds to the myosin, right, the myosin clefts open, right, and it is separated from the actin. The second step, ATP is hydrolyzed, right? So during the hydrolysis of ATP, it has ADP and the phosphate, and the myosin head is moved from this actin. It is moved slightly further toward the Z disc, and it is attached to the another F unit of the actin, and then in the state 3, phosphate is released, and because of this phosphate, the cleft is closed and it is released from here, and quickly the ADP is also released, and this force move back the myosin head to the original resting stage, right? And because there are many myosin heads in the thick filament, so it's not only just one myosin head in the thick filament. There are like so many myosin heads in the thick filaments. So at any given moment, so some are bound to the thin filaments. So this because of that, it prevents the thick filaments from slipping backward when an individual myosin head releases the acting subunit to which it was bound, right? The thick filament does actively slide forward past the adjacent thin filament. This process, coordinated among the many sarcomeres in the muscle fibers, bring about the muscle's contraction, right? So this was the muscle contraction. So this is how the muscle contraction is done. But the interaction between the actin and myosin must be regulated so that the contraction occur only in response to appropriate signals from the nervous system. Correct? Like it's not like your muscles is contracting by itself. Like there are some signals are sent to the from the nervous system to the muscles, and at that moment only your muscles are contracted. So it should be regulated, right? So the regulation is mediated by a complex of two proteins. So the regulation of muscles contraction is regulation of the contraction is done by two proteins. So which are that two proteins? That is tropomyosin and troponin. And troponin. So these are the two proteins, or the complex of two proteins that regulates the muscle contraction, tropomyosin and troponin. So tropomyosin binds to the thin filament, blocking the attachment site for the myosin head groups. So what the tropomyosin does, so the tropomyosin does, what it does, it binds to the thin filament. It binds to the thin filament, that is the actin, right? And what it does is blocks the attachment site for the myosin head groups. So tropomyosin to the thin filament, right? And what it does by binding, blocking, it blocks the attachment site for the attachment sites for the myosin head. So this is how it regulates the muscle contraction. This is the tropomyosin. And the troponin is a calcium binding protein. The troponin is so this is a calcium binding protein. A nerve impulse causes release of calcium ions from the sarcoplasmic reticulum. So when there is a nerve impulse, that can cause release of calcium ions from the sarcoplasmic reticulum, and the released of this calcium ions binds to the troponin, that is another protein ligand interaction, and that causes conformational changes in the tropomyosin troponin complex, exposing the myosin binding sites to the thin filaments. The contraction follows. So what's the process? So a nerve impulse causes calcium 2 ions from the sarcoplasmic, calcium 2 ions from the sarcoplasmic reticulum, right? And the release of this calcium that binds to the troponin, that is the another protein ligand interaction. So the released calcium binds to the troponin. And this is also another protein ligand interaction, and that causes conformational changes to the tropomyosin troponin complexes, right? And what happens, that due to this exposing the myosin binding sites on the thin filaments. And the contraction follows. After that, the contraction is done. This is how the muscle contraction is regulated. Right, so the working skeletal muscles requires two types of molecular functions that are common in proteins: binding and catalysis. Right, and the actin myosin interaction, a protein ligand interaction like that of the immunoglobulins with the antigens, is reversible and leaves the participants unchanged. So when the ATP binds myosin, however, it is a hydrolyzed to ADP and phosphate. So myosin is not only an actin binding protein, but it also has an ATPase and enzyme. All right. So this was actin myosin and the protein ligands functions, and this is how the muscle contracts. All right. Thank you guys for watching. We'll continue with this protein function, and the next topic we'll discuss about the enzymes. All right. See you guys.